Mitosis. Let's begin with that. Basically, one cell splits into two identical daughter cells. In mitosis, when a cell divides into two different cells, both the cells have identical genetic information. Wait, but how does this happen? That's why I'm here, to teach y'all about some DNA replication. First, let's talk about the basics first, like what the purpose of DNA replication is, and then we'll get to the more important and juicy stuff. Let me set this straight. All organisms go through DNA replication. The purpose of DNA replication is to produce two identical copies of DNA molecule. DNA replication is essential for mitosis. Did you know that DNA replication takes place during the cell cycle during the synthesis phase? No, you didn't. So I taught you something. That's a pretty good question, not gonna lie though, like for real. So, where does DNA replication occur? DNA replication in a prokaryote occurs in the cytoplasm. Cytoplasm is a thick solution that fills each cell up to the cell membrane. The cytoplasm is mostly made out of salts and water. However, in a eukaryote, a plant cell, it takes place in the nucleus. So, how does DNA replication happen? There's primarily 4 different steps to it that I'm going to explain. The first step in DNA replication is for the DNA helicase to unzip the double helix by breaking the hydrogen bonds between the nitrogenous bases. This is what makes DNA replication a semi-conservative replication. Semi-conservative replication means that during DNA replication, the 2 strands of nucleic acids separate. The second step of DNA replication is for the primase to place primer on the separated strands of DNA. It places the primer on 5'. Primase essentially tells the DNA polymerase where to start building the second strand of DNA. The third step in DNA replication is to start replicating the original strand to build the second part of the double helix. However, the DNA polymerase doesn't exactly copy the original strand. It makes a strand with different base pairs that will actually complement each other. For example, how the base pairs guanine and cytosine complement each other, and how adenine and thymine complement each other. You can see in the illustration up above, it shows how the DNA polymerase is making base pairs that actually complement each other and that are not identical. DNA polymerase builds a new strand in the 5' to 3' directions. This means it moves along the old template strand in the 3' to 5' direction. The 2 nucleotides don't actually go the same way. They're actually anti-parallel, which means one goes from 3' to 5' and the other one goes from 5' to 3'. Whenever DNA replicates, one of the strands is always continuous and the other one is discontinuous. Continuous means that the primase only needs to put down primer one time and the DNA polymerase can just continuously build and it doesn't need to stop. This is called the leading strand. Discontinuous means that the DNA polymerase is building the other way from the leading strand and that the primer needs to be placed constantly by primase. This creates Okazaki fragments. The discontinuous strand is also known as the lagging strand. But the Okazaki fragments can't just stay as fragments. This is where the 4th and final step of DNA replication comes in. So, how do the Okazaki fragments get back together? This is where ligase comes in. Ligase basically glues together the fragment and makes it so that it's one long strand of DNA. The final outcome of DNA replication is 2 identical strands of DNA. Thank you so much for your time. I hope you give me a good grade on this.